So what's gene regulation and how do genes get regulated? This is sort of what we did. And again, this is big picture, things. We have our genes, our DNA, and we used to think it went DNA to RNA to proteins, done. So if you got the gene for brown eyes, you were going to have brown eyes. But that didn't explain what happened in things like cancer and what happened to how do our hormones get turned on and off all the time, and how do our other cells go on and off all the time. None of that got explained. So what they found is that process is much more complicated. And at every step in the process, we have ways of turning it on, turning it off, slowing it, stopping it, changing it in whatever way, shape, or form. Okay? So if we think about our process, our big picture, DNA to RNA to protein, well, we can regulate how genes get expressed at the DNA level. And that's when, how do we know which genes get which, remember our chromatin gets tightly coiled, which chromosomes get uncoiled to start through transcription? That's regulation. So we can regulate at that level. We can regulate with, for instance, methylation or epigenetics. Getting a methyl group will change which genes get expressed at which times. So we have the regulation at the gene level. We then can regulate at transcription. Which genes get transcribed and at what rate it happens. So how do we know which genes get open and how fast does it happen? We have activators, we have transcription factors that, again, change all those. Now we've made, so that's transcriptional. Now we've made RNA. Well, in RNA, we now can process RNA. We can cut out the introns and splice the exons, and we can do that in a whole bunch of different ways. So we regulate the genes. We can then add on a 5' cap and a poly A tail. That's regulating the genes. So those are the basic ways of post-transcriptional thing, and then we have to get the RNA out of the nucleus. We can stop RNA from getting out of the nucleus. If it can't get out of the nucleus, it's not going to be able to be transcribed or translated. Okay? Then we can relate it at the translational level. So now with the RNA, with the ribosome reading it, well, some RNA lasts a short period of time. You just make it and then you're done. Then it gets broken up. Some mRNA lasts a long period of time. So the length of time we let our RNA go, the amount of time we let our ribosomes go, what we do with the mRNA. So again, we have all this non-coding RNA, it doesn't make proteins, it goes back and affects our DNA. So all of these different things after, during sort of translation we can affect. What happens after we've made our proteins? Well, we can have some proteins that remain inactive till we activate them. We can put some proteins in the Golgi apparatus and the endoplasmic reticulum till we modify them. We can use epigenetics to methylate the proteins and change them. So we can even affect how much, what happens to our gene expression, what proteins are produced afterwards. What if we don't export them from the cell? What if we do export them? What if we only make them at certain times? So again, just like your house, all the electronic devices in your house can be controlled by different things at different times. Some are on, some are off, some are on timers, some are on light sensors, some are on motion sensors, some are worked by a thermostat. These are all the ways that are what we call gene expression. Making of proteins can be regulated. Does that make sense?
Yeah.
So it's just sort of big picture, and if you think about, oh, what are the steps in protein synthesis? How can I fix them or change them? Good. All right, what else?